Hello and welcome back to the Computational Genomics Lab. Today we're going to be plotting TPM values and learning a few different ways to take averages and standard deviations. Last time we did a grep, calculated the average and a standard deviation, and we're able to plot it that way whenever you have replicates. And today we're going to learn how to turn those columns into rows with a function called melt or pivot longer. There's lots of different ways to do that. Yeah, I think this will be another complete lesson in how to be plotting when you have multiple replicates and focused on TPM values. So let's get started. We're here on the Boulder Rinn Lab GitHub, masterclass lessons, and we are now on 07 and 04 TPM plots. You can copy this into an R markdown file in your version of RStudio, whether it's local or on a HPC. And so let's get started. We're going to need these packages. We're going to need to install Reshape. Go ahead and do that real quick and load it in. And again, I'm going to have a lot of more information in these documents. So you can see through the video what's happening, get a feel for it, and then go back and read what's going on here. So as we've been doing, we are going to load the same. I'm going to make sure I'm in the right working directory. Lessons 07, perfect. Set is working directory, nice. Okay, so I'm going to load in our TPM results. And we have our average and standard deviation values there that we made last time. And we're going to load our DE-seq, which is mostly that filtered res df with all the p-values and log-fold changes from DE-seq. So between those two, you really have everything you need. Is a TPM file, an accounts file, run DE-seq, and you're good to go. Okay, so first thing I just want to point out that we've been indexing into columns now using this cache sign. And you can see here that what if you had just labeled your columns by numbers like 0, 12, 24, 48, 96? Well, if you had wanted to go in to your average and standard deviation values here, we have 0 and then underscore average, 0 SD, 12 average. There's a digit there. And so let's say we wanted to go into 96 underscore average in that table. It's not going to work because that's an integer and it's actually thinking about it as a number. So what you can do instead is just put a single quote around it. I know the quotes are hard to remember, but you just try different versions, but in this case you have to have it embedded in quotes so that it knows its character. So we did this before, but we're going to do a histogram of our average and standard deviation TPM values for the 96 average, and we'll set our x-lim and y-lim and breaks here. Okay, let's take a look at what we got. Nice. Okay, so we see that in the 96 time point, most of our data is under 200 TPMs. And again, a lot of them are really low. So those can be important genes. A lot of people cut off with a TPM of greater than 1. But just so you see the distribution, it's really just this distribution and then everything else. And there's very little representation out further than that. Okay, so now we're going to do some ggplotting of our TPM values. And so we're going to do like we did last time, is we're going to take those average and standard deviation value column names, which is 0, 12, 24, 48 average, and we're going to make that an object called time values. And now the time points, we're going to go in and grep those column names. We're going to say names, average, and standard deviation values. If we did that, we'll see if you just run that part, you get the column names essentially. And so we're going to index into those column names and grep anything that starts with that has average and then names of average and standard deviation values, which is going to be the 48 SD. And so it's going to grab just the averages here and not the SDs. Is really all we're telling grep to do, is grep anything that has average in it, and that won't be our standard deviations, it'll be just our averages. Okay, so we'll run that. And then we're going to make an average and standard deviation values 2. So let's see here, average and standard deviation values. Our gene names, gene IDs are in the meta column here. And so we're going to shift those in here so we can start to get access to them. And so we'll do simply doing row names to column, and we're going to call that new column gene ID. And so now when we look at our average and standard deviation values in file 2, we can see our genes have moved into a new column called gene ID. Okay, so now we're going to do the fun part. We're going to melt the data. So this takes a long time to get your head around, at least it did for me. But you'll get there. And essentially right now all of our data is across columns. And it's very hard for a computer to grab multiple things in a column and do something to them. We did do that with grep, but typically you would make each data frame stacked as rows, and then you can group by and grab things as rows instead of grabbing them as columns. So that's what melt, pivot longer, those things are going to do for you, is take your wide data, make it long, so it's actually easier for the computer to grab comparable things. Okay, so to do that, we're just going to use the melt function, and that's in the parentheses, we're going to take our average and standard deviation values, and the measurable variables are going to be time points, and the value names are going to be gene ID. So basically what we're going to do is for each gene has all these things, we're going to say, instead of making that all the way across, make it for each gene have one value all the way down. Okay. Again, it's tricky, and I highly recommend doing a question mark melt and looking at, this is a parameter that's required. So we're saying what we're going to measure is the variables is our time points up here that we just made, which is all the average TPM values, and then the value's name is going to be gene ID, so that each gene is now going to have its average across each time point, and we'll have 4 rows per gene instead of now all the way across. Okay, take a look at that, play with it. It took me a minute here. So now if we look at this melt, it's 109,000 rows, and if we sort by gene ID, we're going to see that each one has 5 entries for each time point of its TPM value. So instead of being one gene with all the values in columns, now it's one gene with each one in a row, and we'll have the values here, and also which time point was retained in there. Okay, so now let's go. This is really just doing the same thing, but using pivot longer. So we're going to take the same average and standard deviation values to this one, and all the difference is really here is we move the gene ID in, so that way when we melt it, we have the gene ID to make multiples of instead of just one, as they are in this data frame currently. Okay, let's clean up some of these files. Okay, so really doing the same thing. We're going to take that file we were just looking at with our gene IDs as columns. We're going to pipe it into pivot longer. And we're going to say for columns, that's time points, we made the object up above. Take all of those and the names to time and values to TPM, which are going to be the new column additions after we turn each gene from one row into multiple rows. Okay, so again, just really the same thing, and how to do it, same length, same number of observations, and we can see here that we ended up with the same result. We're going to have the time and now TPM measured here, whereas in our melt, we had it as a variable name, what the average value was. Okay, so let's go back. So that's, again, practice it, look at it. What did it look like before, after you melted? What were the hardest part for me to understand is like the values to and from, and really you're going to take the gene ID as your main thing you want to make multiple rows of, whereas currently it's only one row. But get a feel for the nomenclature for melt versus ggplot. But now what we can do is simply let's take the melt one. So we'll do ggplot melt df, which is now the longer one. X-axis equals value. So if we look at that, the value is actually the TPM at this time point. and then we're going to make a histogram with 50 bins. We're going to facet wrap on variable, which is each of the different time points, so 0, 12, 24, 48, 96. Scales free, Y, X, lim, and our theme that we just made last time. Okay, cool. So now what we just did very simply, just by melting the data, we could just put it right into ggplot. And then now that we had that time as rows, each different time point as a row for each gene, we can actually get the distribution of TPM values for every time point, which is what we have here. Cool, so we have 0, 12, 24, 48, and 96, and they all look identical. Even though we did scales free, it said, you know, hey, it looks the same to me. So that's great. That's a good first check. There's nothing weird about any of our time points in the genes abundance we measured in TPM. Okay, so you could also do it this way with your pivot longer. It is exactly the same. It's just that it has different, instead of variable in that column, it's now time. Just a little tweak just so you can start to understand how we took something and then made it longer. But really that's just going to make the exact same plot. Okay. And we can also do this by taking the original file and doing a summary, and we can see what the min, max, these kind of things, just to point out how important summary and table are for peeking through your data. Okay, so now we have our sig genes, and what we're going to do is we had this filtered res df here that has 918 genes in it, and we're going to unique it now so we can just look at the values of our significant genes as one representation for each gene. Okay, so sig genes is we're going to make it as a data frame. We're going to use unique on our filtered res df gene ID column, caching in there, and then the names of our sig genes is going to turn into gene ID, whereas currently it's not there. Okay, so let's run this line first. I should actually make a space here because they're two separate things. Okay, so we're just making a data frame of our sig genes, and here we go. There they are. That's the names. And again, we got that funky column name, and that's all this is going to do is saying take the names function for sig genes. So we're going to pop into the names, which is just that one column, and we're going to put in gene ID instead of that long, complicated thing. And now if we look at it, we see it's just gene ID. Simple way, lots of different ways you could replace the column name. I prefer this way. Okay, so now we're going to do an inner join where we take our average and standard deviation values for our sig genes, and we're going to merge. Inner join is just another form of merge. And so we're going to take our average and standard deviation values 2, which I believe is now in long format. Nope, it's not. So we have our 0 average, 12. All we did here is move the gene ID in, and that's what we used to melt previously. So we're going back to the wide format, and we're going to merge it with sig genes. This is, again, kind of like we could take sig genes and square bracket this in that the way we've done it before. I'm just using inner join here as a different way of doing it. Okay, so sig gene TPM, 665 genes, and now we have the average and standard deviation only for our sig genes. And oh yikes, I don't know why this got all jumbled, but this is the same thing I was saying you could do before. I'm not going to actually do it. Is you could take that data frame that has all the values and say take the ones in the gene ID that are in the sig genes ID as well. So that'll give you the exact same result. You can run it just to make sure you see that there, sig gene TPM, 665, and it all looks the same. Okay, so now we're going to add in our gene ID, or gene name. So right now we just have gene ID, so we're going to just, again, learning different functions to do the same thing, is merge sig gene TPM, that's what we just made, with G2S, and they're going to merge by gene ID automatically. In fact, I think the console will tell us that. No, it didn't tell us what it merged by, but if we look at sig gene TPM, we see now that this one has the gene name, and there's H19. That gene's gonna keep coming back. It's actually the first long non-coding RNA ever discovered, but we'll get more into that later. Okay, so we've just added the gene name, not a big deal. And now we're gonna remove the gene ID. So we made our sig genes with the gene name and not the gene ID. Could have done it with both, but we're just gonna get rid of that gene ID, which is the long string number that we saw before. Okay, so now let's pick our favorite gene. This is one of the ones we looked at before. It changes a lot. And we'll say this is a gene that's up, and we'll make a data frame that just pastes in that name here with the C is like cat. The names for that column, we'll call it gene name. So gene up, again, I should have had a space here so it doesn't look like they're connected. Gene up, gene name, there we go. Okay, all the same kind of moves. So now we're going to get its TPM. So we're going to go in now and index instead. We could do the merge, we could do the inner join, those other ones we did before. But now we're just going to take our sig gene TPM data frame, square bracket index, and say, give me the gene names in the gene up gene name, which is just one value. And now we should have like a row for this. Yep, so now we can see the 0 average, TPM, standard deviation. And again, you can see it's less than 1. So this gene would have normally been kicked out of our analysis. Oh, here it's above 1, but it's pretty significantly changing. Okay, so now the row names here are going to be, we're going to change that. So currently they are just 624. So we're going to change that and say, let's make this the same as the gene name. So we're just changing that one row name here. And now it's got its own name. Cool. And then we are going to remove the gene name from that. So where's gene name? It's at the end here. So now that we've labeled the row in meta, we can get rid of this value here. So that's kind of a typical thing of add a column in so you can merge or join or filter and then take it back out after you've put it into meta as a row name. Okay, getting rid of that column there. Now we can make a line plot. Let's do that for just this one gene. Okay, so we're going to grep into our names of GM, blah, blah, TPM, and we're going to get anything that says underscore average. So if we look here, we're going to get the 12 average, 0 average, 24 average, and really what that's going to prevent us from doing is grabbing any of these standard deviation values, and we'll grab those separately. So we could do grep quotes SD, and it would grab just those. So it doesn't have to be a perfect match, it's just looking for it in the column names. So if we look at names here, again, it's just the column names. You can see them here, and it's going to grab anything that has average and keep the values as true. And we're going to do the same thing for standard deviation here. So now we have an average and a standard deviation. The names of the columns at the very least here, 5 for each. And now we're going to plot it. Okay, so we're going to make a data frame to plot. So we can take this data frame and function, and we're going to make a column called time, and as numeric, we're going to change that underscore average to nothing in the average columns object up here. So essentially what that's going to do is change this 0 underscore average is going to go away, and it's just going to be 0, 12, 24, 48, 96. So a cleaner way of plotting it. We're going to have another column called average where we're going to unlist. So it's basically going to go in and not string split them, but break them apart into individual pieces. And we're going to go into our TPM and take index into our average columns. So we're only going to get things that match this string here. And then we're going to do the same thing. So right now they're kind of all connected, and it's just going to unlist them so they're separate objects, and the same thing for the standard deviation. So these two, we are going to take these this object we just made up above, the SD calls and the average calls, and we're going to go into this GM TPM here, and we're just going to grab the values for the averages and then separately for the standard deviations. And then after we've grabbed them through the grep, we're going to make them individual objects with this unlist function. Okay. So now we have our plot df in here. So now we have for this one gene, because we only indexed in there for that GM9045, that we have its average and standard deviation. And this looks ready to plot. Again, you can kind of see it's in long format. And that's usually the way you're going to want to plot. So we're going to take our plot df, which we just looked at. X and y are time. X is time, y is the average TPM. We're going to make a line plot. We haven't done that yet, but this is just going to make dots with the line connecting them. We're going to have error bars. So this is in the statistical layer. We can use gm_error bar, basic ggplot function, and say the axes are y minimum equals average to standard deviation, and average plus standard deviation, and the width is 0.2. So these are really just making the error bars. And it's important to say at each point I want the average and the standard deviation. That's what this is doing for the minimum, setting it, and the maximum is, they're taking the average and standard, the top of the error bar and the bottom of the error bar is what we're making here, and how wide that'll be, theme, and then labels, just to add some flair to our plot. And so now you can see what you would kind of typically see. It's always important to plot your error bars, and we can see this gene is, I don't know why the hours seem wrong. This was 12:24. Okay, so we might want to change the scale on the x-axis here because it's a little confusing, but this is 0, 12, 24, 48, and 96, and we can see at 12 hours it actually went from a tiny value to nothing, and then it peaked at 48 hours and then went back down a little bit. Okay, not the best looking gene, but this you can see is very different than those sort of dot plots we were showing before. So let's do it for the whole data frame. Okay, so we just did it for one. Now we're going to use that same logic and do it for all the genes, or multiple genes. So here I have the logic flow again sort of written out to go back and think about it, because I do think this class is particularly tricky. We're doing a lot of new things by melting data, and creating error bars are not the easiest things to learn right away. Okay, so we're just going to make a data frame of our genes of interest. Here I grabbed the gene ID. I could do the gene name. It wouldn't matter. And so now we have this little genes of interest data frame, and it's just their gene IDs. And now we're going to grab them grab our TPM values from that. So we're going to say row names of TPM filtered. Now that was that data frame where we took anything that didn't add up to 1 out, and so that's got all of our sig genes in it. Yeah, so it's like the filtered resdf, but for TPMs it's just our sig genes. And we're going to take that, and we're going to take the row names, which are the gene symbols for all the 998 things, in our genes of interest gene column. Those are the rows we want, and we want to keep all the columns for this. Okay, so data frame to plot genes. Now we can take a look at that, and we see basically all we did is take our gene ID and grab all the TPM values we had before, and we're in that wide format, and we're going to do the whole pivot and get things done. But first we need to move the row names back in, because currently they are in the meta. We're going to move those in so we can melt by them or pivot longer. And this is really a super common practice. You take a wide data frame that's an output of something, turn it to plot it. Okay, so now our df plot genes has the gene IDs as a column. That's all we just did there. And so we're going to pivot longer. We could do the melt function, and we're going to take these df plot genes data frames, so the genes we're interested in plotting. We're going to pivot longer. The columns we're going to remove gene ID that we moved into meta. So we're just, instead of doing it as a separate step, doing it in this function. Names to time and replicate, so that's going to be the new column name after we take each gene and give it multiple values as rows. The column, it's going to retain the time value and name it, and the values are from the TPM itself. Okay, so let's see what happens when we made that longer. We wrote over it. I should have called this longer. But now what you can see is there's many more rows in here, and we have the TPM column that we labeled and the replicate. So we remember where it was originally before we made it longer. And then we are going to take time and replicate and sort of do that string split we did to get the time and the replicate value. So we're going to take that now long format. For each row, we're going to separate the time replicate column. So this one here. So last time we did string split with the underscore. This time we're going to say separate into wild type, time, and replicate. So they're going to make 3 new columns with each of these values, and we're going to separate by the underscore, and then merge them, and then remove that wild type we made. Okay, sort of some nice tidyverse. So if we look at it here, so now we have this really handy thing of the time point, the replicate, and the TPM. And really all we did is just split those names apart with the separate function. And now we are going to convert these to numeric values. Currently, our DF plot genes are in number space. This is also one of the hardest things about bioinformatics to me is, are you an integer, number, character, logical? And you'll always know here because it'll say it what the column names are. So these are all character and this is a number, but we want to get it into integer numeric space here. And so let's do that to our time column, which is currently a character. So we want to convert that to a number, like our TPMs, and we can see that just happened there. Cool. So now that's something the computer can think about as a number. And now we're going to group by, and previously we calculated the average and standard deviations before. In this case, we have each individual replicate value, and we can use group by in our DF plot gene. So like here, we have 5 rows with this, we have lots of rows because we have all 15 replicates in here. So we have 15 rows for the same gene. So group by will come in and say, give me all the values that are the same in this column. And then I can do things to the time column, like take the average or standard deviation, and same with TPM. So last time we did a grep across, calculated it, and repopulated the table. This time we've taken just a raw table, made it longer. We're going to group them by their gene names or gene IDs and do things while they're grouped together. Okay, so group by is here. We're taking that same data frame we were just looking at, and we're going to group by gene ID and time. So we're doing a double group so that one gene has 15 values, and inside there, each time point has 3 representatives. So it's going to be a 15 group and then 5 3 groups within that 15 of 0. So this is like sort by in Excel. You're sorting by first the gene name, then the time point. And then once we've done that, we can summarize the mean TPM. We can say what that's going to equal is the mean of the TPM. And the standard deviation is going to equal the standard deviation of the TPM. And these will be 2 new columns we have in that data frame. Okay, let's look at our DF plot genes. Nice. So now we have the mean and the standard deviation. And we have it 5 values now instead of 15 for each time point. So we really did the same thing we've done before. I prefer this way, but I just wanted to do it a few different ways to kind of get familiar with it. And now we're totally ready to plot. So we've got this data frame we just looked at, X is going to be time, Y, same as this one, mean TPM. And that's now a column name, mean TPM. And we're going to make a line plot, same error bars, labels, and everything, and basically making the exact same plot. We did a facet in here somewhere, facet wrap by gene ID. That's slightly different than last time. So, oh, we didn't add the theme paper white either. So this is the default sort of theme. And if we look at these, well, this one looks really good. Very low error bars, and it's going up. So what is this gene, I would say, is a good one to look at? Is it H19? Don't know, because we use the gene ID, so it's harder to know. Okay, so I've given you some exercises here. I think it's most important on this one to go through these exercises. But what we did, you could see we did a lot of things the same. We took a wide thing, we turned it, we summarized data into a smaller data frame to plot. What if we made a function that just said, make me figures? And you put in what data frame, and it made all those figures. And you wouldn't have to do this ever again. You need to know how to do it, but you wouldn't have to do it again. So we're going to have a bonus class on this one. If you go down, we're going to actually make a function that does all this, but go through it on your own. And how do you make your own function? We've been using R's base functions, but we're going to have a bonus video for this class to make a function, and that function will be to make a figure of any gene you're interested in. So let's go do that.